Right, in 26.3, we're going to talk globally about how we track disease, how we recognize patterns, and we'll talk about finding what we call the index case or patient zero, and then talking about databases, things like that. All right, so in May and June of 2011, we had 6 U.S. residents develop these strong cases of bloody diarrhea. 5 of them developed life-threatening hemolytic uremic syndrome, and eventually one of those people died. Hemolytic uremic syndrome is bleeding and inflammation in the kidneys, and as we see, it can be very life-threatening. They found the organism there, and all of them were the same serotype of E. coli. We call it O104H4. So this is normally an E. coli that doesn't produce the Shiga toxin. Normally we find it in the enteric tract, but it's not a pathogen. And in this case, though, it did have the Shiga toxin gene, so it has acquired that. In this case, that is what caused the hemolytic uremic syndrome. The other interesting thing is when the data was collected, these 6 cases didn't cluster to any single location in the United States. They're widely distributed. We have 5 different states that they occurred in. At the same time, in Europe, there were a large number of people who died from hemolytic uremic syndrome. So by June, there were 3,200 cases and 35 deaths in Germany and France. The organism in this case was the same odd type of E. coli O14H4 with the Shiga toxin. If we don't have public health organizations that share data and on incidence and prevalence and things like this, we would have not realized a connection here. But there were indeed connections. When they tracked it back, right, they started asking questions. Okay, there must be some common thing here. The clinical labs shared the data, and the CDC identified they were all the same strain, and then we start tracking travel. In the case of the U.S. patients, they had all recently traveled to Germany, where they had been infected. And then we go, okay, they've all gone to Germany, and we have clusters in Germany and France. So where has this come from? Well, they tracked it back to bean sprouts from a single farm in Germany. So we worked with the data from the incidents, and we start correlating things. And this is where it's really important to have reporting and databases. Local physicians are required to notify the CDC of specific cases of infectious disease, things like anthrax, botulism, what else we got on here? Cholera, syphilis, typhoid fever, measles, Ebola, obviously, Giardia, things like this. So there's a whole list of diseases that they're required to report to. But what happens when you have something that's an unknown? We don't know what's causing it. Well, that's when we report what we call syndromic surveillance or symptomatic surveillance. We say, hey, we've seen 5 people that have this set of symptoms or this, syndrome, which is a set of common symptoms. That can help us identify new and emerging pathogens. Once we see that occurring, we start to trace this back, and we look at how people have come into contact with one another. So this is finding our patient zero. And in the lab, we did an exercise that somewhat simulated this, but in reality, you're looking at history and contacting people, but it's very, very difficult to do this in reality. And trying to identify cases from these people. So in this case, we can see clusters of individuals, and there are some that are in yellow that we will talk about in just a moment. Ideally, we can contact trace and quarantine positive cases to hopefully cut transmission. Or we can identify common links between all these people. Say they all went to the same farm or ate the same sprouts. So we're going to look at this case of severe acute respiratory syndrome, or SARS, which is caused by a coronavirus. And in 2003, an outbreak started. So we tracked the SARS outbreak back to a single individual, a woman from Singapore, who was staying in a hotel in Hong Kong. She is patient number 1 here. You will notice off of this, we have numerous cases occurring. But you can see there's certain individuals in here that infect large numbers of people. So she infected 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 different people. Whereas this person didn't infect anyone, and this person infected one more person. That person infected 3 other people, right? So here's what's going on. Okay, so she was in Hong Kong and got infected. We can't trace it further than that, although there are some thoughts that maybe there was a doctor who had been working in rural China who had brought it to this area in Hong Kong. So she traveled home from Hong Kong to Singapore. But then she went to the hospital when she got home because she was not feeling well. She had fever, headache, and a dry cough. In the hospital, they did a bunch of tests and she was negative for any known pathogens. So they placed her in a general hospital ward without any sort of barriers to infection or control measures or things like this. Okay, we have an unknown. That makes it difficult. They sent some samples actually for electron microscopy because they suspected maybe it was a virus. Most hospitals are not equipped to do electron microscopy. They're very specialized instruments, and finding new viruses is difficult, right? We can't grow them, so we have to identify them by microscopy. In this case, they identified what we called the SARS, severe acute respiratory syndrome coronavirus. And they got that from nasopharyngeal samples. So just like SARS-CoV-2, our COVID-19 causing virus. So 6 days later, they identify this virus and go, uh-oh, we got a respiratory virus, but it's too late. She's been in that general hospital ward, and by this point, she infected 20 other people, so even more than is on here. This includes other patients, staff, and visitors. So people are dispersing, right? Patient 6 in this case was a nurse who then went on to infect many more people, including patient 35, who was a diabetic with heart disease in the same room, who then went on to infect more people. Number 6 also infected this person, who then infected this person, who went on to affect a lot of people, and this person as well. You will see the colored circles are what we call super spreaders. Super spreaders are people who transmit the infection to 10 or more individuals. That is our definition of a super spreader. And there are various factors as to why that can happen. Maybe they have a higher viral load, maybe they're shedding it more, coughing more, not observing proper hygiene things, or just interacting with more people. All of these things can happen. This outbreak led to over 1,000 deaths worldwide. We quickly instigated things like quarantine and masks to help contain SARS, and it was easier to contain because of some of the genetic properties of it. It didn't spread quite as easily as the new virus that we've had recently, SARS-CoV-2, which has been much worse as we've seen. It's very easily transmitted. So this is one of the reasons that global surveillance is so important. Knowing that SARS outbreak was occurring helped us kind of screen people that were coming to the United States. Unfortunately, not every country has things that are necessary, like modern clinical laboratories or efficient case reporting systems, or even effective political systems that can, detect this, limit outbreaks, do things. If you have a country that's in the middle of a civil war, it's very difficult to organize this information and get it out, particularly when the government is in shambles. So developing countries have limited resources, and that can make public health difficult. This is why the CDC and the WHO send teams out there of scientists, medical professionals to go and help in these areas. Not just because we worry about what might come back to us, because a lot of people travel to the United States, but also just to help, right? Saving lives is important, and we can do that through public health. All right, so we talked a little bit about tracking outbreaks. It comes from contact tracing and asking questions, collecting data, and organizing it in databases. Obviously, finding patient 0, the original person, is important in containing the spread, because then you can get all the cases that are coming out from that. And then rapid responses that can prevent outbreaks from becoming pandemics are obviously necessary. All right, that's it for 26.3.